Thank you all for coming. I was going to wait a few more minutes for a couple of teachers that I know are going to arrive, but I know the students have tight schedules, so I think we'll get started. And I'd like you to welcome you to Oceans Alive to hear the science fair winners from Falmouth High School and Falmouth Academy. This is always, I think, a very exciting time to see students put their results together, and we always schedule it before the state science fair. And I'm always impressed every year how talented and insightful the students have become. So they've decided what order they're going to present. And since Kevin has to leave early to study for French, he's going to go first. And so our first speaker this afternoon is Kevin Huang-Lin, who's a student at Falmouth High School. And he'll present his findings on the evolution of marine cyanobacteria in the Red Sea. Kevin?
Hello? Yeah, okay. Well, good afternoon, everyone. My project this year is a continuation of my research from last year, and is titled The Evolution of Marine Cyanobacteria in the Red Sea. I performed my work down at the Marine Biological Laboratory under the direction of Dr. Mitch Sogan in the Josephine Bay Paul Center for Comparative Molecular Biology and Evolution. And for this project, I've been fortunate to work with Dr. Anton Post, who is a visiting scientist from the Hebrew University in Israel, and he really helped guide me on this project. So, as my title suggests, I studied the evolution of marine cyanobacteria, and my project focused on two specific species of cyanobacteria, or families, Prochlorococcus and Synechococcus. And these organisms are both unique and significant in the in global climate processes today, as well as global ecosystems. First of all, they are both photosynthetic prokaryotes, so they use sunlight as a source of energy. Furthermore, they are two of the smallest known photosynthetic organisms on this Earth. Prochlorococcus has a diameter of just 0.5 to 0.7 micrometers, while Synechococcus is just slightly larger, with a diameter of 1 to 2 micrometers. However, despite their small size, they do live in an extremely wide range of habitats, anywhere from 40 degrees north to 40 degrees south of the equator, and can live within the water column from the surface of the ocean down to 200 meters deep, where sunlight is limited, but apparently enough for these organisms to survive. So due to this wide range of habitats that they can inhabit, these two organisms together account for 2/3 of the ocean's photosynthetic processes. And today, with the issue of global climate change, that of course is a very significant factor to potentially reducing carbon dioxide levels in the ocean, in the atmosphere. So this map just illustrates some selected strains of Prochlorococcus to illustrate their range, anywhere from the Indian Ocean to the Pacific Ocean to the Mediterranean and the Red Sea, which is where I studied, where I focused my research. And the Red Sea in particular is located 28 degrees north of the equator. And what is specific about this environment is its unique biogeochemical cycles. Annual mixing and stratification causes nitrogen stress from April to October within the water column. During these months, the limited mixing within the water column allows heavier nutrient sources to fall towards the bottom of the Red Sea, and therefore leave the organisms that inhabit the top photic layer, such as Prochlorococcus and Synechococcus, with significant levels of nitrogen stress, in which they lack nitrogen. And therefore, these organisms must adapt to these lower levels of nitrogen in order to survive throughout the months of April to October. So the objective of my project was to determine, through sequential, functional, and phylogenetic analyses, if Prochlorococcus and Synechococcus have evolved to the unique nitrogen-stressed conditions and other environmental conditions within the Red Sea, and if they therefore differ from strains of Prochlorococcus and Synechococcus found in global bodies of water in other environments where such environmental stress conditions are not present. And my approach to this research involved three major steps. First, I sequenced and assembled the DNA of 4 strains of cyanobacteria from the Red Sea, Prochlorococcus marinus, strains 8380C9 and 13A3, as well as Synechococcus 4320C2 and 4320C3. I unfortunately wasn't able to travel to the Red Sea to collect these samples myself, but my advisor helped me by bringing them over from Israel. The second part of my research involved a determination of the functions of the cyanobacterial genes after sequencing. And finally, I compared the DNA between the Red Sea cyanobacteria as well as between the organisms from the Red Sea to organisms from other locations. And specifically for my research, I chose a strain of cyanobacteria, Prochlorococcus, that was isolated from the Pacific Ocean, which is an area that doesn't exhibit the same environmental stress conditions as the Red Sea. So the first part of my research was a sequence and assembly, as I mentioned before. I grew plasmid-containing E. coli cells from a stock and purified the DNA to obtain the isolated DNA that I needed for sequencing procedures. I used automated sequencing machines to obtain the DNA sequences and assembled the DNA using computer software. So I identified the base pairs within the genetic material that I was given and assembled them into continuous sections of DNA. And from this, I was able to successfully sequence the 4 cyanobacteria that I was researching. The Synechococcus DNA for those 2 strains were assembled into full-length sequences. So for all of the genetic material I was given, I was able to come up with a complete section of DNA. Whereas for the Prochlorococcus, the DNA was assembled into multiple pieces or multiple contigs. This was just a result of the sequencing procedures. There were some missing sections in between. But for the purposes of my research, that was enough to make significant comparisons. And I'll talk about that more in the next part. In total, I sequenced approximately 208,000 base pairs of DNA, or an average of 52,000 base pairs per organism for each of the 4 strains that I analyzed. And these photos are just from some of the sequencing and computer assembly procedures. So the second part, after obtaining these DNA sequences, was a functional analysis in which I tried to determine what the genes identified in my organisms actually did, how they actually functioned in the organisms in their natural environment. So to do this, I referenced the National Center for Biotechnology, or NCBI database, to look for matches between the genetic material in the cyanobacterial DNA and the genes from other organisms that have been previously sequenced and uploaded to the database. And based on the most significant matches to organisms already within the database, I was able to give functional assignments to the cyanobacterial genes I was working with. From this, I was able to identify 192 total open reading frames, which correspond to probable genes, among the 4 cyanobacteria that I was sequencing. It was an average of 48 genes per organism. And what's interesting about the top matches within from the cyanobacterial DNA to the database is that 96% of the Prochlorococcus genes matched most closely to other cyanobacteria, and 100% of the Synechococcus genes matched most closely to other cyanobacteria. So based on this, you can see that even though these organisms live in such an isolated environment such as the Red Sea, they do share significant, extremely significant genetic similarities to other strains of cyanobacteria living in other bodies of water. Further, 26 Synechococcus genes were given functional assignments related to nitrogen assimilation and metabolism. And you might recall that the nitrogen stress levels within the Red Sea would make it necessary for these organisms to have some sort of adaptation to those environmental stress conditions. And these genes are possible adaptations that these organisms have shown. However, between the two Prochlorococcus organisms that I worked with, there was only one gene given a function related to nitrogen assimilation and metabolism. And of course, that's a very significant difference, 26 versus 1. And I'll elaborate on that point in just a bit. So based on the functional assignments that I obtained for these organisms, I proceeded to the final phylogenetic analysis, in which I tried to observe how these organisms have adapted to nitrogen stress conditions within the Red Sea. And to do this, I created visual maps of each genome section based on the functional assignments that I came up with. And I identified conserved genes, or genes that were similar between the organisms, and use them to align the sequences as a basis for comparison. And by comparing these genes, I tried to determine how and to what degree the organisms in the Red Sea have evolved to nitrogen stress conditions and how they therefore differ from the Pacific cyanobacterial strain that I was dealing with. So the next few slides show the layouts that I created from the genetic material I sequenced. These are the 2 Prochlorococcus organisms and the 2 Synechococcus, and each of them shows the genes laid out. Each of those block arrows represents one gene. You can see the functional assignments pointing to each of the genes. And the genes with the blue highlights indicate genes related to nitrogen assimilation and metabolism. So between the 2 Synechococcus organisms, this slide and this slide, you see there are a number of teal-colored genes, whereas among the Prochlorococcus, there's simply one here and none in the first one. So using these layouts that I created, I was able to come up with this alignment here. And the genes indicated by the dotted box there are all represent euporphyrin C methyltransferase, or COB-A, which is one gene that I identified between all 5 of these organisms. And I used that as a basis for aligning the 5 organisms that I was working with. Among the 2 Prochlorococcus organisms, the C9 contact 39 and the 13A3 contact 28, you can see a number of genes match up in terms of positioning and direction. And also among the Synechococcus genes, there are a number of genes that match up in terms of direction and positioning. So what did I learn from these alignments here? The 4 organisms that I sequenced were successfully aligned based on the presence of the euporphyrin C methyltransferase in all 5 organisms. And furthermore, the vast majority of the nitrogen-related genes were identified in the 2 Synechococcus organisms, with only one nitrogen gene identified in the sequence of Prochlorococcus 13A3. So what can I conclude from this work that I've done here? First, that the average sequence length of each cyanobacterial genome represents approximately 3 to 5% of the entire genome for each of the organisms. This was enough material for me to work with for this research. To sequence the entire genomes would be almost unrealistic for a project in this time period. But since I was able to come up with that significant identification of the COB-A gene in all 5 sequences, I was able to align them and compare them to look for evolutionary divergences. And the 95 to 100% similarity of top hits of genes in the cyanobacterial species that I worked with to other cyanobacteria in the NCBI database show that there are extremely significant genetic similarities between the Red Sea organisms and organisms in other environments. However, the lack of nitrogen assimilation and metabolism genes in the Prochlorococcus organisms suggests that these organisms lack nitrogen utilization genes, so that they represent organisms that are unable to metabolize and assimilate nitrogen compounds other than their normal nitrogen compounds of ammonium between the months of April to October, and are therefore left in significant states of nitrogen stress, whereas the Synechococcus are well adapted to the nitrogen stress conditions and can use nitrate and nitrite compounds from April to October when ammonium is not available. So this provides a puzzling situation here, that the Synechococcus have somehow genetically adapted to the Red Sea conditions, whereas the Prochlorococcus seem to have not adapted to these nitrogen stress conditions. So to further study this issue, it would be necessary to do more genotypic investigations, possibly further sequencing the Prochlorococcus genomes to see if the nitrogen genes have maybe been translocated to other sections of the organism's DNA, which is unlikely but is possible and could be done through either genetic probing or by sequencing the entire genomes. However, it would also be interesting to do some more phenotypic investigations in terms of field work by looking to see how these organisms, Prochlorococcus specifically, have adapted to survive in the nitrogen stress conditions of the Red Sea when nitrogen is not available. So to do this, it could involve taking a field trip out to the Red Sea to take a look at how these organisms work in their natural environment. And it is a possibility that there are, I know from my research that there are heterotrophic bacteria living within the Red Sea that do produce small amounts of ammonium from other nitrogen compounds that could sort of spoon feed nitrogen compounds to the Prochlorococcus organisms in some sort of symbiotic relationship during the periods of nitrogen stress during the half of the year that ammonium is not generally available. So in my research, I should give thanks to Dr. Mitch Sogan and Hillary Morrison, who helped me very much, as well as Dr. Anton Post, who provided the samples for my research, Ms. Christine Brothers from Falmouth High School, who supported me through the 4 years that I've been doing research down in the Woods Hole community, and the Oceans Alive and WHOI Sea Grant programs for allowing me to present my research here today. Are there any questions?
Any questions for Kevin? So do you hope to travel to the Red Sea with Dr. Post at some point?
Yeah, I'd like to.
Or somewhere
That would be,
from college?
yeah, that would be fun.
So you get the wet side of
Yeah.
the investigations.
I have to find a visa to get over there first.
Yes, how long have you been working on it?
I've been doing this project for 2 years at the MBL, but my research started my freshman year with some other projects.
Any other questions?
In determining what each gene did, was that through the data bank that you got that information? You were able to tell that some were nitrogen related?
Yeah, through the NCBI database, I looked for similarities. So some of the genes matched up almost 100% in terms of base pair alignments to genes that have been previously sequenced and identified within the NCBI database, even though they're from other organisms. Based on those significant genetic similarities, I can infer that the functions would be the same between the organisms as well.
In the other 6 months, is there any nitrogen stress the opposite way in the other 6 months? Maybe in excess or?
Well, for my project, I focused on the specific lack of nitrogen from April to October. In the other months, I believe it's a sufficient amount of nitrogen for those organisms to survive, not necessarily an excess of nitrogen.
I was just going to ask where you got your foundation in genetics. Was it a combination of your coursework at the high school and the assistance with folks from MBL?
Well, before I started my research freshman year, I took a course over the summer, and that gave me a brief introduction to genetics, and that was a subject that I was interested in pursuing. So I found Dr. Sogan's lab down at the MBL and have been working there since then.
So in college, are you going to continue to study biology?
Yeah, I hope so.
Any other questions? John?
So here's maybe a hard question. Maybe not. The inference that there is a difference in function or not, based upon the presence of similar genes, may not tell the whole story. Have you thought about how the genes that may be similar in the Pacific and the Red Sea organisms may make the same protein, but the protein sequence may be slightly different, and therefore the function may be different in those different places?
Yeah. Well, based on the research that I've done, it shows what kind of genetic similarities. Of course, the protein differences the proteins could have different functions, specific functions with the organisms, and I think that's one major reason to continue my study past this genetic basis and look more on the bigger picture to see how the organisms actually interact with their environment.
So your phenotypic study may show.
Yeah.
Great.
Well, thanks, Kevin.
Thank you.
Our next speaker this afternoon is Caroline Cotto from Falmouth Academy, and the title of Caroline's project is Feeling Crabby: How Serotonin Affects the Aggressive Behavior of the Invasive Asian Shore Crab. So, Caroline?
So can you hear me? Okay. The title of my project is, as she said, Feeling Crabby. And just to start generally, the brain is able to instruct its electrically activated neurons to secrete chemicals known as neurotransmitters. One of the most famous is serotonin. And it's a monoamide generated from the essential acid tryptophan, and it's commonly known as 5-HT. Serotonin is most centrally located in the raphe nuclei part of the brainstem, which you can see there. And it's proven that in vertebrates, and specifically humans, high levels of serotonin are known to incite feelings of euphoria and relaxation while also controlling things such as appetite and sleep. And having abnormally low levels of serotonin can have detrimental effects such as bulimia, insomnia, and most of all, severe depression. Depression affects more than 19 million people in the United States today and is often controlled by antidepressant drugs. But however, in invertebrates, serotonin has a completely opposite effect. So I began to wonder how increased serotonin levels would affect the behavior of the Hemigropsis sanguineus, which is an invasive species that was brought to the Atlantic by ballast water in 1988, and it's characteristically. So I began to wonder how increased serotonin levels would affect the behavior. And it was therefore hypothesized that increasing the concentrations of serotonin would provoke more aggressive behavior and dominant postural stances. Although this experiment dealt with new research, the procedure was modified from an experiment done by Dr. Kravitz, who's a leading scientist in serotonin studies who works at Harvard, and he did experiments with serotonin in lobsters. So, working off of his experiments, I took 68 male Hemigropsis sanguineus that were collected by the Marine Biological Laboratory, and I had to prepare a serotonin solution that I could inject into the crabs. So serotonin hydrochloride powder was added to buffered seawater in order to create 1 millimole of serotonin solution, and this stock solution was diluted with buffered seawater in order to create 4 concentrations, 5 micromoles of serotonin, 25 micromoles, 50 micromoles, and 100 micromoles. And since students aren't allowed to handle syringes, Dr. Alan Kazarian of the Marine Biological Laboratory helped me to inject the crabs with 100 microliters of serotonin solution at a soft, fleshy spot at the base of their claw, which you can kind of see it's right below the first arms after the claws. And this spot was chosen because it allowed the serotonin to enter directly into the bloodstream of the crab, since they only have semi-closed circulatory systems. And after injection, the crabs were left alone for 30 minutes in order that the serotonin could be fully exposed. When the interactions began, 2 Hemigropsis at a time were placed in a divided miniature aquarium where they were allowed to acclimate for 3 minutes before being allowed to interact for 7. The divided tank was to ensure that the behavior of the crabs was actually caused by the serotonin and not a change in the surroundings or the close proximity of an opponent. So during the 7 minutes of interaction, the crabs were observed, notes were taken, and key moments of the fight were video recorded. Then observation notes and a scoring system adapted from that of Dr. Kravitz was used to give each crab an individual score of aggression on a scale of 0 to 3. And I had 4 types of fights. There was 5 control fights in which 2 untreated crabs were allowed to interact. The second type of fight involved a serotonin-injected crab and an untreated crab who were allowed to interact. And there was 5 of these fights for each concentration of serotonin. The third type of fight involved 2 serotonin crabs interacting, and there was 5 of these fights at the 25 micromole concentration at 100 micromole concentration. And the final type of fight was between an untreated crab and an injected crab. However, the injected crab had been left to be exposed to serotonin for more than half an hour in order to see if time would have any effect. So once I collected my data, I set up this main graph, which, as you can see here, compares the concentration of serotonin to the intensity of aggressive behavior. So first and foremost, after looking at these graphs, the aggressive behavior of the Hemigropsis seems to correlate linearly over the 4 increasing concentrations of serotonin. At 5 micromoles of serotonin, the aggressive behavior average was 1.25. At 25, it was 1.54. At 50, it was 1.75, and at 100 micromoles, it was 2.15. When a line of best fit was calculated for these averages, the correlation coefficient was an almost perfect 0.99134, meaning that basically the more serotonin, the more aggressive. Although this can be trusted pretty confidently, it's important to note that there is some statistical scatter with standard deviations ranging from about 0.66 to 0.72. And the data showed that the results of this experiment clearly followed the invertebrate model and crustacean pattern of aggression that was outlined by Dr. Kravitz in his work with lobsters. And another way to explain this central data is that the serotonin in this experiment may be acting as a neuromodulator instead of a neurotransmitter. The difference is that a neuromodulator enhances or reduces the activities of neurons, so serotonin may be regulating the aggressive behavior and not just plainly initiating it. Furthermore, as mentioned previously, it is known that there's a substantial difference between serotonin's effect on vertebrates and invertebrates, yet it's extremely hard to decipher why there is such a difference. It's easy to get a basic overview of what serotonin does in invertebrates. Basically, control of aggressive behavior lies in the decision-making neurons of the central nervous system and in the large neurosecretory neuron, which is called A15HT. High levels of serotonin are said to increase the firing rates of these neurons, causing them to release more serotonin into the synapses between the behavior-controlling neurons, therefore sending more signals to initiate an aggressive behavior. It's not as easy to find the exact effect of serotonin on specific neurons in vertebrates, so there's still no tangible answers as to why having a backbone makes such a difference. It may just be a developmental accident. A last conclusion that can be made from this graph is that for their size, the H. sanguineus were just as aggressive as the lobsters used in the Kravitz experiment. The only main difference was that the crabs replaced the lobsters' tail flips with kicks. And normal aggressive behavior involved aggressive stances, claw grasps, kicks, blocking, and attempts to overturn. So, moving to the time graphs, because of a clear lack of pattern in the data and overlapping error bars, which aren't shown here, the exposure time to serotonin did not appear to be a significant factor in the aggressive behavior. And similarly, fighting injected crabs with each other does not seem to have made a significant difference in the aggressive behavior, which may be a result of when you fight two injected crabs, they're better equalized, and so the aggression seems less severe. And like all experiments, this experiment had some sources of error. Mainly the crabs were not weighed and then injected according to their weight. So if I were to do this experiment again, I would definitely want to inject them to get a more accurate amount of serotonin for each crab. And different crabs of different sizes fought each other, which I didn't really have a lot of control over, but if I were able to do this experiment again, I would like to find opponents that were more equally sized. And then some of the crabs were missing appendages. As you can see here, this one is missing one of its legs, which didn't seem to make that much of a difference, but could have affected the data. And finally, although the control in this experiment was meant as a control, it was not the ideal control because the crabs that were used as the control should have been injected with buffered seawater before being allowed to interact to maintain consistency. Some future studies for this experiment might fight native species versus invasive species, injecting one or both with serotonin in order to see if increased aggression in invasive species is actually a critical threat. Another future study might see at what level of serotonin aggressive behavior declines, if there is such a level. And a third future study might see if other neurotransmitters or neuromodulators have opposite effects in vertebrates and invertebrates. So the relevance of this experiment is twofold. It first lies in its attempt to investigate the role of neurologically active chemicals and their evolution. And it's also meant to understand the way the nervous system is affected by imbalances of neurological substances. And this is the A15HT neuron that I was talking about before that actually helps to control the aggressive behavior in invertebrates, specifically crabs and lobsters and other crustaceans. And secondly, the environmental relevance. This project provides preliminary evidence that it's important for antidepressants to be kept out of the water and that runoff containing such drugs be controlled. I don't know if you guys have been following the news a lot, but in March there was an article by the Associated Press on pharmaceuticals in the drinking water, and over the past few years there's been a lot of articles on how antidepressants are in the water. And although these are trace amounts, because it's already known that this H. sanguineus is an aggressive species, and this experiment shows that even trace amounts can affect the aggressive behavior, these amounts could cause it to exhibit increased aggressive behavior, and which could lead to the depletion of native species and aid in the infiltration of the hemigrapsis, which could further affect the food web, although not directly exhibited in this experiment. So in conclusion, the results of this experiment show that the hypothesis was fully supported, and I'd like to thank my biology teacher, Dr. Amit, and my mentor down at MBL, Dr. Alan Kazarian, and Dr. Edward Kravitz from Harvard Medical School, who was my email correspondent, and my family. So.
Anybody have any questions for Carolyn?
What?
Why did you choose that type of crab?
Just because it was so new to the area and it's really taking over. If you go to the beach and lift up a rock, you're most likely to find a Hemigrapsis sanguineus. And a lot of people don't actually know what they are because they blend in really well and they are very resistant to environmental effects, and they don't have a lot of natural predators. So.
How do you quantify the aggressive behavior of crabs? Do you measure how much they move within 5 minutes? Or?
I had a chart from Dr. Kravitz which outlined the aggressive behaviors that were considered a level 1, level 2, or level 3. And after doing some basic observations of the crabs, I modified that chart with changing the lobster behavior into crab behavior. And then I used that as a basis to determine how aggressive each crab was.
Did any of the crabs that you were studying go through a molt?
Not while I was studying them, but I do know that crabs molt, so it's possible that they did after my use of them.
How many crabs did you use?
I used 68 in total. So, they were all male.
I noticed that you proposed to look at Carcinus manus, the green crab.
Yep.
Did you know that the green crab was introduced? by
Yeah,
Dr.
that's why native was in quotation marks.
And it's been a very aggressive and successful colonizer.
Yeah, and actually there's a lot of data out right now that suggests that the Hemigrapsis is overtaking them already. So, yeah.
Maybe if you can find some cancer crabs who are truly native.
Yep.
I think the comparison with even the three species.
Do you think you would have found different results if you were working with female pollinators?
I'm not exactly sure, but males tend to be more aggressive. That's why I use them. I don't actually really know. I would suspect probably not at the moment.
So are you going to continue this?
I don't think so, but
Something brand new.
yeah. I like working a lot with animal behavior. My project last year was on butterflies, so I'll have to find something new. No.
Any other questions? Thank you very much. Our next speaker this afternoon is Neil Forrester, and Neil will be presenting his project, Differentiating Random Sequences with Strongly Typed Genetic Programming. Neil is very familiar to this stage.
As you can see, the title of my project is Differentiating Random Sequences with Strongly Typed Genetic Programming. Real random numbers are a very important thing in modern society because it allows all codes, cryptographic schemes for keeping data secret to work. You can't keep any sort of a message you want to send or any sort of data from prying eyes if you don't have some method of inserting an unpredictable or random element into that data to make it so that other people can't read it. And as such, there's been a lot of work in making random numbers. Some of the easy ways of making random numbers are dice or coin flips, but those are slow. So if you have a larger capital investment, you can make random numbers a lot faster than that with radioactive decay and measuring that with Geiger counter, or with a CCD chip from a digital camera in the dark. But all of these require specialized hardware that the average internet user might not have if they want to, say, send their credit card number over the internet via secure connection to buy something from an online retailer. So that's why, so problems like that are why pseudorandom number generators were created. And what they do is they basically take a formula of some sort and create numbers that are as unpredictable as possible from that formula. And the advantage of this is that you can do it very, very quickly on a computer, and you don't need any sort of special hardware to do it. But the disadvantage is they're not actually going to be random numbers because they were created by a formula. And as John von Neumann, one of the pioneers of coming up with pseudorandom number generators, said, anyone who considers arithmetic methods of producing random digits is, of course, in a state of sin. But he himself was in a state of sin because he came up with one of the very simple and early generators called a linear congruential generator. And what it does is it takes some number in a sequence and it multiplies it by a constant, adds a constant, and then divides by a constant and takes the remainder to get the next number in the sequence. And it has various non-random properties, but it's very easy to implement, and it's a good starting point for testing simple schemes using it. And to test a random number generator, a pseudorandom number generator, to make sure that its results are sufficiently random for whatever you're doing, there are a lot of different tests that you could do that test various statistical properties of the sequence produced. They test things like the distribution of the numbers, whether one number occurs more than another, or whether certain sequences of numbers appear more often, or whether there are periodicities in the sequence, whether you have a repeating pattern of any sort. But all of these test specific, these all test a particular property of a pseudo-random sequence that somebody thought of that might make it non-random. And these have there's a certain disadvantage in that because it implies a human bias in what using techniques like this gives a lack of creativity in trying to decide whether a sequence is predictable or random. Changing gears completely now, there's a certain variety of computer programming techniques called a genetic algorithm. And the only similarity between a genetic algorithm and real genetics is that a genetic algorithm is based on the principle of evolution and natural selection. And what it does is it tries to find solutions to complicated problems that perhaps even the person posing the problem doesn't fully understand. And it's one of several AI techniques, artificial intelligence, that allow a computer a certain measure of creativity or expanding beyond the capabilities most people might associate with it, such as pure working with a single formula to produce a definite result. And the way a genetic algorithm works is it begins by initializing a population of possible solutions. And these solutions are some mathematical representation of a solution to a problem. For example, if you were trying to come up with a computer program for determining whether a sequence of random numbers is random or not, like I was doing, then the population would be a computer program. It would be a set of computer programs, each one being a possible solution to the problem. And that's generated randomly at the beginning of the genetic algorithm. And a whole bunch of these programs are then evolved as a real population evolves to a certain extent. The objective function is the next step, and what it does is it decides and gives a numerical score to how good any particular solution to the problem is. What it does is it tests out the solution and decides on a number and that gives a definite, this is a very good or a not so good or this is a really bad solution to the problem. Then once you have a number for each member of the population, then you can move on to the death phase, and that is according to some algorithm. I used a particular one, which I'll get to in a bit. You decide, based on the scores produced by the objective function, which members of the population should stay in the population and which should die off. And you remove from the population whichever bad ones you select. Then you have the breeding phase, where it takes some number of good solutions to the problem, measured by the objective score, and it combines them in some way and produces new combinations of their genetic material from the old ones. And this is a process similar to real organisms breeding. And then to introduce new material into the gene pool that undergoes a sort of mutation guided by a pseudorandom number generator, and it introduces new twists or new genes, so to speak, into population in order to allow it to continue its evolution. So what I wanted to do with this project was to create a genetic algorithm capable of evolving computer programs that can tell apart a random and a pseudorandom sequence. And the specific way I wanted to do this was for a computer program created by the genetic algorithm to take a sequence of random numbers and assign it a score, separate from the objective score. That's saying how good the program is, but the program assigns each sequence a score. And then for that score, for that program, you can determine whether, through some testing, whether that, what the cutoff should be for this score saying this sequence came from one random number generator or this sequence came from another. And it doesn't really matter how the program internally decides whether a sequence of random numbers came from generator A or generator B. All that matters is it produces a number, and that you can use this number to decide whether the sequence of random numbers or pseudorandom numbers you gave to the program is from one generator or another. And this allows you to do several things. Firstly, if there is a way to decide whether a certain random number sequence came from one generator or another, then you know that at least one of those two sources of random or pseudorandom numbers can't be random. Because if you can tell them apart, then there is some difference. And you should not be able to tell apart random data from random data. And another interesting thing you can do with this is if you have a collection of computer programs developed by the genetic algorithm to work with different pairs of random number generators, then you can take a sequence of random numbers and pin down with a certain degree of accuracy where that sequence of random numbers came from, how it was generated. So in my specific genetic algorithm, I used a technique called genetic programming. And that's using a genetic algorithm to write a computer program. So the population initialization phase of the genetic algorithm consists of generating a tree of numbers like this, which represents a computer program, and I'll get to how it does that in a bit. And it uses a pseudorandom number generator to decide on the exact structure of the tree and which numbers to place where. the objective function then takes the population of genomes, each of the trees it represent being a genome, and it runs the objective function on that and produces a list of scores. And the way it does that is first it takes the tree of numbers and translates it into a tree of operations. And each of these operations takes in numbers from the base and feeds numbers higher into the tree. For example, let's look at this addition node right here. Taking a close look here, we can see that it has, as inputs to it, below the node, the numbers 8, 4, and 2. They happen to be constants in the tree. And the addition node will add them together and output 14. And that will be passed up to the next highest node in the tree. And that will then perform whatever operation it does on the number 14 and other inputs it gets, and so on up the tree until you have a number coming out of the top, which is the return value of the program. That's the score the program is assigning to a sequence of random numbers. And the input operator is a special one because it allows the program to get input from a random sequence. For example, if this is a random sequence here that you're testing with the program, then this input operator, it has a 7 as input, so it finds the 7th number in the sequence, which is 36, and it outputs that into the tree for it to operate on. And in this way, the tree can take input from the random sequence it's working with and decide what the score for the sequence should be based on that. And the operators return numbers up the tree and eventually out the top, and that's the final score. But there's a little problem with a scheme like this, and that's where strong typing comes in. What strong typing does is it allows the tree of numbers here, the parse tree, to not just operate on numbers like 3 or 4.5 or 47. What it allows you to work with various types of data. So, for example, a list of numbers or a 1 or 0, just allowing you to specify on or off, yes or no. And it also eliminates problems like this. For example, you have an operator that compares the sizes of two inputs, and that would produce, in normal computer usage, a 1 or 0. But suddenly here you've got a division operator. And now you're trying to divide by either a 1 or a 0. Division by 1 doesn't do anything, and division by 0 produces an error. Strong typing eliminates this by having each operator in the tree have defined input and output types. Say this operator can only take in an integer, or it can only take in a list of numbers. And it eliminates problems like this because you no longer have nonsensical operations going on. For example, you couldn't say whether 3 is greater or less than the list of numbers 7, 8, 1. That makes no sense. You can't perform such an operation. And so strong typing dictates the structure of the tree and provides constraints in its creation and throughout the rest of the algorithm, saying that it has to be syntactically correct. You can't have a tree that doesn't make sense. And this allows you to do various interesting things. It allows you to push around large chunks of data in the tree rather than just discrete numbers. And it also allows you to take a vast amount of input from a random sequence, which is a very useful thing when you're looking for very subtle patterns. And the objective function, overall, what it does is it takes a bunch of random sequences from one source of random numbers that you're trying to optimize the genetic algorithm with, and a whole bunch of random sequences from another random number generator. And it runs all of these sequences through the program that it's testing at the moment. And it comes up with a value for each random sequence. And then it performs what's called an ROC analysis. And basically what the ROC analysis does is it determines a threshold for whether a particular number indicates one thing or another. So in this case, what we've got here are a frequency plot with the frequency along the y-axis and the number returned by the program being tested along the x-axis. And the red line could represent the frequency for a particular program when it's of its output when it's operating on sequences from one random number generator. And the green line could represent the outputs of the program when it's operating on sequences from another generator. And the ROC analysis decides to put a line in that says, for all numbers below this line, it is as likely as possible that that number came from the program running on one random number generator. And on the other side of the line, it is as likely as possible that the sequence, that the number there came from the program operating on a sequence from the other random number generator. And you can calculate two values called the sensitivity and specificity. If you were doing something like medical testing with an ROC analysis, then there'd be a defined which is which for sensitivity and specificity. But in this case, they're assigned arbitrarily. What they are the sensitivity is the fraction of points from one generator, the red points, that lie on this side of the line versus divided by the entire number of red points. So you have a fraction between 1/2 and 1. And the specificity is the fraction of points from the other generator that lie on the other side of the line. And since these are each a fraction between 1/2 and 1, 1/2 indicating perfect overlap, and you have 1/2 and 1/2 on each side, and 1 being perfect separation, and the two groups of values don't overlap at all, then you can come up with an objective score. And since you're trying to maximize both the sensitivity and specificity, it makes sense to make the objective score their sum. So you have a number between 1 and 2, 1 being very bad and 2 being perfect. The depth phase then takes the scores between 1 and 2 for each member of the population, and it orders the population based on the score. So you have all the low scores down here and the high scores up here. And it then eliminates the bad ones. It decides these ones aren't very good and just destroys them from in the population. Then the breeding phase will take the best ones of the population and breed them. Obviously, the real population in the genetic algorithm is much bigger than 6. This is just an example. Usually it's more on the order of 60 or 100. And for each pairing of trees that it decides to breed, what it does is it randomly selects a node in each tree, for example, this one and that one, and then it swaps those nodes and everything below them, so that you now have two new trees that are made up of the structure of the old ones, but are different. And you repeat this process many times, and you eventually have what is very similar to genetic crossover when real organisms breed, when you have the DNA from one parent for a while, and then you switch and you have DNA from the other parent. The mutation phase then takes it does essentially the same thing within the tree, swapping around material. And also, it can sometimes just pick a node and destroy everything below it and randomly generate a new tree there. And so you have completely new material being introduced into the population as well. And then it does this twice for every pair, because you produce two children for every pair, and it reinserts these children into the population, keeping the original ones. And after repeating the cycle many, many times, as many generations as possible in an hour in my case, because my computer can't possibly handle a normal evolutionary timescales, for example, 1,000 or 1,000,000 generations. I usually ended up doing around 60 generations. It comes out with the final result. It just decides, all right, this is long enough, I'm stopping here. And it takes the best member of the population, the member with the highest objective score, and it then says, this is what it's got. And this is the best it was able to do within those 60 generations. Now, there's a little problem that often occurs in the results, and that's called overspecialization. And what overspecialization is the data that was used to evolve the trees. I couldn't, due to reasons of practicality, I couldn't have completely new sets of random and pseudorandom data for every generation. I had to have a pool of random numbers, of random sequences that I could draw from. And that caused the genetic algorithm to get used to these sets of sequences and produce programs that were tuned to look at these ones in particular and not general characteristics that would occur in sequences from the generators in general. So to combat this, I had a separate set of random sequences that didn't enter into the evolution at all, and I would match them up. Not match them up. I would run the objective function on the best member of the population, but with the new set of random sequences, so that any coincidences or anything that it encountered the last time around in the sequences that would allow it to say all the sequences that have a 3 in the third spot should be over here, and all the sequences that have a 17 in the 8th spot should be from the second generator. Those coincidences would not occur in the other set of sequences because they just occurred for no reason. But any that occurred because there was something fundamentally wrong with one of the generators would remain. So to illustrate how easily this can occur, I got 100 numbers from a real random source, and I arbitrarily divided them into 2 groups of 50, and then divided those groups of 50 into sequences of 10 numbers each. And I just looked at these 2 groups and thought, how can I decide whether a sequence is from group 1 or group 2? So I looked at the second number after a while and realized that if it's above 100, then there's an 80% chance that it's from the second group. And if it's below 100, there's an 80% chance it's from the first group. And there's no real reason it should occur. If I get another 100 numbers, this isn't going to happen again. It just happened to occur this time. And if one of the programs evolved by the genetic algorithm happens to look at random coincidences like this, it's going to get a very low score when the objective function is run on a new set of random data. Some of the results I got, this is an overspecialized solution. It's evolutionary progress. Along the x-axis, you have the generation number, how far through the evolutionary process it is. On the y-axis, you have the objective score between 1 and 2, 2 being very good. Now, this was very overspecialized. You can see that it started evolving, and the red line traces the best member of the population, and the blue X's indicate the members of the population at that point in the evolution. So, for example, at the 10th generation, you've got a couple of very good members, but a lot of bad ones. So, as you can see, evolution progressed, and it seemed like the objective scores were getting very high, and the population was evolving to meet these new standards. But the check score was very, very low, very near 1, indicating that all this was really useless progress because it wasn't looking at any of the real coincidences in the data between a random sequence and a pseudorandom sequence. In this case, that was generated by a linear congruential generator. A good solution, however, it shows much the same pattern of evolution. It starts low, eventually gets higher, but the check score was much higher, around 1.5, indicating approximately a 60 to 70% chance of correctly identifying the source of a sequence of pseudorandom numbers. These are all the results I got in a condensed format. The green are the check scores. That's the real indicator of how a given solution performs. And the blue are the original scores. And it shows that there's very little correlation between the original score, what was produced, what the genetic algorithm thought it had, and the check score, what it really had. This is an interesting result. Last year, when I did this without strong typing and with several other differences in how I did it, I ended up with all essentially useless scores below 1.2, but one absolutely excellent score up around 2.0. This is a not necessarily better, but different and more consistent, at least, result with the strongly typed genetic programming over without strong typing. About a third of the sequences had a probability of more than 60% of classifying a random sequence correctly. That third of the programs, though, didn't have any really excellent numbers, but it had a lot of mediocre ones. And some potential applications of this are basically testing a random number generator to decide if it's useful in whatever cryptographic scheme you're thinking of using, and deciding what random number sequence, deciding what generator created a random number sequence that you might have stolen from some other source, stolen from someone else. If you're trying to break their code, you could use the genetic programming to decide whether their random number generator is a possible loophole, a possible chink in the armor of their cryptographic scheme. And that's becoming very important nowadays, since codes that rely on the security of the algorithm itself for using the random numbers become more sophisticated and much harder to break. You now have to look for other methods of cracking somebody's encryption scheme. And I'd like to thank my mentor last year, Dr. Hanyamant Singh, and my mentor this year, a professor at MIT named Uname O'Reilly. Dr. O'Reilly helped me get a lot of reference materials and pointed me in some new directions and towards strong typing in particular, which gave me the interesting results I got this year. So are there any questions?
Thank you. Any questions for Neil? Fascinating.
Yeah?
Neil, how close is what you have generated this year to any applications? Very simple.
You could apply this in a form similar to what I've got it in to testing random number generator, but it's not a really excellent. It's not really perfect for that role yet. It's still got a lot of work to be really useful. It is useful for looking at a very simple random number generator, such as the linear congruential generator. But for more sophisticated means of generating random numbers, it is not at all effective in deciding whether a sequence of random numbers was generated by a real random source or a very good pseudorandom source. And that could indicate one of two things. One, that it needs more work, or two, that pseudorandom number generators are very good and there's no need for concern about their security. And I personally prefer the second option because that would give a lot of peace of mind to the entire population of the world. Yep.
You mentioned real random number source that you were using. Did you take out those numbers from different places? Did you create it yourself, or did you bring it from somewhere else?
I used two different sources of random numbers. I don't personally have any hardware that's able to efficiently and quickly generate real random numbers, so I relied on internet databases or sources available on the internet of real random numbers generated from hardware sources. I did start with 2 sources of randomness, but I discovered a problem with one of the sources, and that's why I had the second source, so that I could do checks between them. I ended up with a source with a lot of random numbers that are real random data, and those numbers came from a device called a J1000KU, produced by the Comskier Corporation. And basically what it does is it has a very noisy electrical amplifier, and it uses it basically listens for the noise in the amplifier and has various filters to produce a random bit stream very quickly. Question there? Yep.
Let's say you just went around to 10 people that you had no idea who they were or anything. Just walked over and asked 10 different people, say, to name a number, and then you took those numbers. Would you consider them random enough?
They could be random, but I would doubt that they would be, because people have a certain bias not to pick certain numbers or to pick certain numbers. For example, if I asked pick a number between 1 and 100, you're very unlikely to pick 1 or 100 because you would perceive that to be non-random. Whereas you would be just as likely to pick 100 as you would be to pick 42 or 84. And so people have these biases to try to randomize the results, which actually de-randomize them. So I would doubt that people picking random numbers would be a very random way of getting data.
From Falmouth Academy, and the title of Maya's project, The Effect of Sleep Deprivation on Cockroach Olfactory Memory.
All right. All right. Hi. Thank you for this opportunity. My research was about the effect of sleep deprivation on cockroach olfactory memory. Memory and sleep are both pretty enigmatic processes. We deal with them a lot in modern life. We need to sleep, we need to remember things, and yet we don't know all that much about them. And what's especially intriguing is the role of sleep in memory formation. Another thing is that sleep deprivation is a very modern problem because of busy modern schedules, sleep disorders. I'm sure you all don't get enough sleep. I never do. Sleep deprivation is really a major problem in humans, and it does affect memory retention, but we're not quite sure how. I wasn't able to work with humans, so I looked for a species of invertebrate that I could test to look at the correlation between sleep deprivation and memory retention. There are only a few species of invertebrates that actually do experience periods of rest or sleep, including fruit flies, bees, and cockroaches. I wasn't about to work with fruit flies, so instead I used cockroaches. Cockroaches are common pest native to the United States. I used a species called Paraplaneta americana, the American cockroach. As you can see, that's the adult and the nymph. They're not really that size, but they're about the size of 2 knuckles. These neurologically, cockroaches' brain setup is not very similar to humans. Their neurons are spread out much more in their body. For instance, they sense light more through their skin than from eyes.
and their sense of smell is picked up on the antenna as well as the feet and the lower part of the head, and not just from a nose like in humans. However, cockroaches are similar to humans in that they also experience periods of sleep, and that they also rely on memory. Their best sense is their sense of smell. They use it to orient themselves and find their way back home. So what I decided to test in terms of memory was their olfactory memory, their sense of smell, and how that was affected by sleep deprivation. So, as I said, that was my goal. I hypothesized that sleep-deprived cockroaches would have poorer memory retention than those who were allowed to rest. For an experiment, I worked with several different studies that I found, some with bees and fruit flies and some with cockroaches, and sort of combined studies of the cockroach olfactory sense and sleep deprivation and circadian rhythm. So cockroaches are trained to associate, I wanted to train cockroaches to associate one odor with a rewarding taste and a second odor with a punishing taste. This is a method that I found in a study by Decker of Vanderbilt University. She wasn't doing anything with sleep deprivation, so with her methods there was a lot of tweaking to do, but she did use two specific odors. She used vanilla and peppermint odor, and I'll get back to that. And what I wanted to do after this training was deprive the cockroaches of sleep for 24 hours and then test their memory retention. Oh, sorry. So the first thing I did was to reset the cockroach's circadian rhythm. Circadian rhythm is a free-running biological clock. Cockroaches, like humans, have a very strong circadian rhythm, and so I reset their circadian clocks, as you can see in the pictures on the left, circadian night and day, so that I could test at their highest arousal state, so that they were always as energetic as possible, moving around, and I could get the most behavior out of them. And like I was starting to talk about, in Decker's study, she found that vanilla was the Paraplaneta americana, that cockroach species preferred vanilla as an odor and most disliked the odor of peppermint. In terms of rewarding and punishing tastes, she found that a sucrose solution was the most agreeable, most preferred taste for the cockroaches, and a saline solution most disliked. So I decided to use those four. And the picture on the top right shows you a little bit of my methods of training. It was very difficult to work with the cockroaches in the way that Decker had done so, because she was working with a lab and assistants enough to be able to isolate one cockroach at a time, show them, I guess, or put them near an odor, and then with a syringe give them a definite amount of solution. However, cockroaches, I found, are not only kind of nasty little buggers, but very much followers. And so it's really difficult to get them to go from one place to another unless you move one, and then they'll all follow, and that can take hours and hours. So it was necessary to train and test the cockroaches in groups of 5, which made doing statistics a little bit difficult. The picture on the bottom right is something I'll get back to, but if you can sort of see, it's a little bit circular. It was one of my training arenas. Those dark things in the middle are cockroaches. They're on a piece of paper. I set the piece of, they like to hide in darkness, so that piece of paper was something in their cage, and it was dropped into the testing arena to sort of get them to run down into the testing arena. So the first thing I did was some preference tests and controls to make sure that the cockroaches actually did prefer vanilla to peppermint and sucrose to saline. They did. And another great thing about doing that was that I was able to see some behavior that would later on help me to quantify memory retention. The cockroaches that approached the sucrose solution would put their feet and antenna into the sucrose solution, move away, and then sit down, bend over, and begin licking their legs. They like the taste, they're licking their legs. Cockroaches that approach saline solution would put their antenna and feet in the saline solution, touch it, shake, and run in the other direction. So they really didn't like the saline solution. And so that event of bending down and licking the legs to taste the sucrose solution was something I called a tasting event. And I would later use that as one of my ways for quantifying memory retention. What I did was I had a group of about 60 cockroaches. I had to eliminate some because they were injured or not adult. I was unable to separate out males and females because I just didn't have enough for replication purposes. And what I wanted to do was test half of the cockroaches, to train half of the cockroaches against their natural preference and half according to their natural preference. So those trained against their natural preference were trained to associate peppermint with sucrose and vanilla with saline solution. Those trained to associate to heighten their natural preference, to go according to their natural preference, were trained peppermint with saline solution and vanilla with sucrose solution. And then half of each of those groups I sleep deprived. The way I did that was I constructed a sort of a shake table, a very loud shake table, using a rock tumbler, which you can see in the bottom photo. And that kept them awake, shaking, loud noises, for 24 to 25 hours. In other studies that I read, other methods of keeping invertebrates like this awake are triggering an automatic predator response by blowing CO2 into the cockroaches' faces. However, that would usually mean isolating each cockroach into sort of a chamber and having air jets at their faces, and that's very complicated. So I didn't do that. And then in testing the cockroaches, after being sleep deprived or allowed to rest for 24 hours, they were placed in groups of 4 or 5 in a testing arena, which was a 30 centimeter diameter round chamber, and 180 degrees away from each other was the peppermint odor source and the vanilla odor source. So the cockroaches were placed in the center and were allowed to wander around for 10 minutes. And I observed them to look for specific behaviors that would let me know whether or not they were having any memory of their training. So one thing that was very obvious is cockroaches would visit an odor source, proximity, nearness to an odor source. Secondly, a standing event. A standing event was cockroaches had a choice to either walk around the odor source or walk right over the top of it. It was a petri dish, small petri dish with holes drilled in the top, so they didn't fall in, but it was easy enough to walk across the top. Sometimes the cockroaches would actually stop on top of the odor source and try to feel sort of closer to the odor with their antenna and their feet to try to smell it better. And so I called that a standing event. And thirdly, as I already mentioned, a tasting event. A cockroach would approach an odor source and would suddenly remember and associate that smell with the taste of sucrose and bend down and begin to lick its leg. And that was the easiest behavior to see because it really bent full around. In terms of visits to different sources, that was a very clear way to look at preference in my controls, but it gave ambiguous results in the actual sleep deprivation tests. So I focused instead on my other 2 criteria, standing events and tasting events. In terms of standing events, the group on the left shows cockroaches trained against their natural preference, peppermint with sucrose and vanilla with saline. And the group on the right shows cockroaches trained according to their natural preference, peppermint with saline and vanilla with sucrose. The brown bars show the percent of the total standing events that occurred on top of a specific odor source before even any training ever occurred, so in the controls. With the group that was trained against their natural preference, the sucrose source would have been the peppermint. So before any training, about 20% of standing events occurred on top of the peppermint. After being trained, 50% of standing events occurred on top of the peppermint, which means that the training was somewhat effective. Then after being sleep deprived, the percent of standing events occurring on top of peppermint went down to about 30%, which means that some of that memory and that training was lost. For the group that was trained according to their natural preference, the sucrose linked odor source was vanilla. So there was already a very high preference for vanilla, as you can see. Almost 80% of standing events occurred on top of vanilla anyway, before any training. After training, that preference was heightened a little bit, up to about 85%. But after cockroaches were sleep deprived, the standing events dropped down to about 70%. So there was some drop, not a huge drop, not as much as in the group that was trained against their natural preference, but a fairly significant drop. In terms of tasting events, there's no way to know about the effectiveness of training, because before being trained to associate a smell with a taste, there's no way that a cockroach would have known to recognize a scent and then associate it with a smell. But cockroaches, again, the group on the left are cockroaches trained against their natural preference. So peppermint is, in this case, is the sucrose-linked odor source. And on the right, the cockroaches trained according to their natural preference. So vanilla is the sucrose-linked odor source. In the group on the left, those cockroaches who were not sleep deprived, about 74-75% of the tasting events occurred near peppermint. Those that were sleep deprived, about 67%. So not really a hugely significant drop. The cockroaches trained according to their natural preference, about 64 or 65% of tasting events occurred on top of vanilla when the cockroaches were allowed to sleep. Those not allowed to sleep, it went down to about 50%, so a bigger drop. And I'm unsure as to why there was just a smaller scale of a more balance in the tasting events occurring with the cockroaches that were being trained according to their natural preference, since it was already a natural preference. However, it may have been due to just the overall exhaustion of the cockroaches. I'm not kidding when I say that a tired cockroach looks like a tired human. It stumbles around, it's total zombie, has trouble walking up on top of the odor source. And it may have been that cockroaches just said, I don't care where I am, I'm just sitting down and resting. And I would have no way of knowing they were resting as opposed to tasting. Also, it may be that the scent of the vanilla and the scent of the peppermint didn't travel at exactly the same rates, or they were sensed differently in the cockroaches. The peppermint extract that I used was oil-based and the vanilla was water-based, and to me as a human, the peppermint smelled stronger, so it may be that a cockroach could smell a vanilla odor on the other side of the testing arena and would just sit down right there and start licking its leg, as opposed to moving closer to the odor source. So what does this mean? As I said, ambiguous results about visits to the odor source meant that it wasn't really a good criteria for looking at memory retention. However, in terms of standing events, training was effective, or relatively effective, and cockroaches did have difficulty retaining training regardless of whether they were trained against or according to their natural preference. In terms of tasting events, sleep deprivation did reduce olfactory memory. Now, due to, like I said, a relatively low number of replications, because I was working with cockroaches in groups and recording their behavior in groups, it was difficult to do much in the way of statistics, and it's difficult, therefore, to say how significant of a decrease there was in memory retention. It may have been a small or a very large decrease. Of course, looking at that impairment relative to humans, it's difficult to say whether the impairment would be the difference for us between a sort of tired memory loss that we could drink a cup of coffee and it would get better, or the kind of exhaustion that means we need to sleep another 14 hours and we can't think at all. In conclusion, within the caveats that I discussed before, there is a correlation between sleep deprivation and memory loss in cockroaches. In humans, like I said, lack of sleep affects memory. Although cockroaches and humans are neurologically different, they may share a common response to sleep deprivation. And the question is, can this similarity be exploited to study the connection between memory, sleep, and functionality in humans? For future research, first thing is procedural errors that just come from working with live creatures, especially not in a lab. Things like the fact that I was working with cockroaches of all different conditions, age ranging between adolescent to adult, both genders, varying health. Cockroaches do fight with each other. One morning they might have all 6 legs, and the next day they have 3 or 4, and there's nothing I can do about that. And they are also very fast moving, so if one escapes, I'm in danger of killing it. Also, like I mentioned, other differences in a range of odors. Odors with the same molecular size, very different molecular size, oil-based, water-based. An odor perhaps that would elicit a stronger aversion from the cockroaches than peppermint did. It seemed to me from my results that the cockroaches didn't really run from or hate the smell of peppermint nearly as much as they were repulsed by the taste of saline, or on the other extreme, as much as they preferred and liked the scent of vanilla and the taste of sucrose. Other research could be about memory to do with different stages in the circadian rhythm. I did read some really fascinating studies about memory retention as affected by the time of training and the time of testing. In humans, that means, do we learn better at a certain time of day? Do we test better at a certain time of day? And of course, like I said, varying conditions and varying organisms. In humans, the sleep cycle and the need for sleep changes depending on gender and age and health. Is this the case with cockroaches and other organisms? I'm not sure. And other organisms, invertebrate and other organisms, who either experience very different periods of rest because of their neurological needs, or just don't need to rest at all. I would be interested in finding out how memory and memory retention are affected by sleep deprivation in those cases. And thanks go to my chemistry teacher, Dr. Johnson, and my family for putting up with the cockroaches, and Dr. Decker, who provided me with a lot of information about her research. Thank you. Rana?
What does olfactory mean?
Smell related.
Perhaps you touched on this at the end, but had you considered depriving them of sleep immediately after their training exposure, but then letting them rest before testing?
Yeah, I considered it was mostly because of my scheduling that, I mean, I was still in school and I was up changing the lights for the cockroaches, and the duration of the training and the testing, it does take a long time, so it's difficult. The Decker study did work on short-term and long-term memory, so they tested cockroaches 5 minutes after having been trained, and 5 hours, and 24 hours, and 48 hours, with varying results. So that was something I had thought about, but it just really didn't work in terms of logistics. Other questions? Yep.
The cockroaches have the biological kind of cycle like us, like 24 hours, which I assume is sensitive to the light.
It is almost a 24-hour cycle. However, they are nocturnal creatures, so their highest arousal state is just after dusk, and then they'll come out and they'll rest most of the day. They're not out for a full, 12 or 24-hour block, simply because they like the night, and of course during the year, the period of darkness changes. They're not working under electrical light the way we are. They don't go to bed and wake up. Other questions?
compare the cultures of American and Belgian cockroaches. And I could not sleep without looking at those cockroaches ever. I had nightmares for weeks. And the Belgian cockroaches are twice the size of the American. So I really applaud anyone who can work on cockroaches. I quickly switched to marine invertebrates.